expecting the final stage of what's coming out, but you just started with a single cell and stopped somewhere halfway in the movie, you will most likely not be able to tell whether it'll become a frog or a shrimp or even a human being. We all started like that, right? We all started at that single cell stage and became us, right? And this, in that brine shrimp, was triggered by the addition of water. You might think I've added water, so I have generated the sufficient condition to generate a flow. Okay? I want to impress upon you now that just adding water is not sufficient to generate a flow. And I'm using this word flow very loosely right now, okay? You all saw this embryo moving about, you know. If you didn't know it was an embryo, you would see that something is moving there. You might say there's a flow of something, there's a movement of something, right? And I'm using the word flow to mean a variety of these things at the moment. And that's what I loosely refer to as this flow of life. And why do I say that just with water, it's not sufficient? And I want to show you that even with water, the inside of a cell can quote unquote jam. Okay? And we'll see why. So here is a cartoon representation of one of the simplest cells that we know, a bacterium called E. coli. It's a bacteria. And if you look at the inside of this cell, what is drawn in these colors are the various proteins and macromolecules that are needed for the functioning of this cell. Okay? You've probably heard very often that 70% of us is water. Or 70% of whatever, some large percent of us is water, right? But if you look at the inside of a cell, you'll be very hard-pressed to find where this large fraction of water is, because it is so packed. So if you look at, so this is a simulation, and 2-scale simulation of a small section of this cell. And so each one of these colored blobs is a protein, an enzyme, a macromolecule, etc. So you really see how crowded it is. Yeah, it's really packed, and things are having to push each other for space, right? And you might ask yourself, if that is the case, how can I generate a flow? If I don't even have space to move about, how can a flow come about just because there's water? It seems like it's not sufficient. Okay? So a small detour into states of matter, because it's important for us to set this idea of flow on slightly firmer grounds, okay? So if I asked any of you what the states of matter would be, you would say solid, liquid, gas, right? If I take water, it will exist as a liquid. It'll be a gas in the form of steam, or it can be a solid because it forms ice, it forms a crystal, right? There's another very interesting state of matter, which Janvi referred to Shrikant Shastri at the beginning of this talk. Shrikant is a physicist at Jawaharlal JNC ASR, and he works on this rather interesting state of matter, which is called a glass. Okay, so a glass, we all have everyday experience of what glass is. So I have glasses right now, right? You all have, some of you have glasses. That's the same glass that we are referring to. And it's a very interesting state of matter because water can also exist as a glass. And this is one of Shrikant's interests in water. How can water exist as a glass? Right? So what's the difference between these states? So if I have molecules or atoms comprising a gas, these molecules are so far apart from each other. They're not feeling each other by any interaction. Their positions are random in space and they're moving about. In a liquid, they're slightly closer packed than in a gas. That's what we learn in school. They're slightly closer packed such that now they can interact with each other. They can feel each other's forces. There are small forces of attraction and repulsion that they start feeling. And because of these forces of attraction and repulsion, what they do is to have some order. They start to form some pattern close to each other. If you go all the way away from this liquid into the state of a crystal, the interactions between them are so strong that they form a periodic arrangement, okay, as in the case of a crystal. But somewhere in between, depending, and this is a very complicated matter, there is this state called a glass, which behaves like a solid, but at the microscopic scale, it looks like a liquid. It doesn't look like a solid. Okay? So we have everyday experience with this kind of a situation. For example, in a traffic jam. Okay? So normally when, of course, in Bangalore, most of the traffic is in this glassy or jam state. Okay? Otherwise, it flows, right? Why is this a glassy state? Because you see, things are very disordered. It's not like in a crystal. And at the same time, it doesn't really move. It's jammed. So could something like that be happening in a physical situation like this? So why do I bring this up? Why do I bring this up? Because you have earlier seen this picture. So you have seen a bunch of molecules sitting inside this cytoplasm, and they're so crowded that they really cannot get past one another. They jam. Okay? So this material inside the cell jams up, and it forms something like a solid. So what causes them to flow at all? Generally, what I want to impress upon you is the fact that there are large-scale flows. So what you're watching now is a movie of one cell, and even though it's very crowded, you see that things flow, right? So what is causing this flow? What is causing this flow is the fact that there is energy that the cell can use and burn, okay? So in a traffic jam, if we were not drivers, we would be stuck there forever. But because we can consume energy and move a bit here, there, we can generate a flow. So even though the material inside this cell is so crowded, if imagine that one of those molecules spends energy and pushes, then the neighbors are forced to move along. And once they move along, you generate some kind of a larger scale flow. Once you have a flow, what does the flow really achieve? It achieves the fact that you transport material from one place to another place. Right? And once you have transport of material from one place to another place, you have the possibility of a reaction that can happen. Right? Once the possibility of a reaction happening is there, that can trigger the process, further processes of life. So, can we turn off the flow in a cell by not just removing water, but by removing energy? Right? So that's what I just told you. I can create a flow, not just by removing water, but also by removing a source of energy. And in fact, people have done these experiments where they look at the bacterial cytoplasm. Cytoplasm is the inside of a cell. It's all the material inside a cell. And what they say is that the cytoplasm has glass-like properties. We've just seen that it's so crowded that it has glass-like properties. But it is fluidized by metabolic activity. What is metabolic activity? It's really just the consumption of energy, right? So what I'll very briefly try and explain this experiment to you. So what you're seeing here is the outline of a cell. And what they do is they put a little molecule in there that glows bright, is fluorescent. Okay? And they can visualize this thing moving. Imagine now that I'm in a traffic jam. And you cannot tell the difference between any cars. You're sitting so far away. But one of the cars is lit up. Right? If this car remains stationary, you'll know that there is a jam. But if it starts moving, you know that it's not jammed. So that's the technique. So they use this fluorescent cell, and what they do is they deplete this cell of energy. If it's not depleted of energy, this little molecule moves around happily. So this blue and red line is the trajectory of these molecules that are moving inside the cell. But as soon as you remove energy, you see that the spot, so the molecule just becomes trapped in one location. Right? So what has happened between this state and this state, here there's something liquid-like or fluid-like, and here this has suddenly become like a solid. Okay? And what I want to show you guys is that when a cell enters this state of dormancy, or this suspended animation or whatever else, it goes from something like that to something like that. It becomes a solid. So removing water or energy initiates the transition to dormancy. That's the statement that I just now made. Okay? But how does the cell know that it's being removed? I mean, water is being removed, energy is being removed. It doesn't. So typically there's some other cue. Right? Something like the pH outside changes, or some other chemical cue comes saying, oh, there's going to be very little energy from now on, and so on, right? And it uses those cues, the cell uses those cues to put itself into dormancy. That's an active process, okay? So it goes from a liquid, and I use the word active liquid, because it uses energy to make the cytoplasm liquid, right? It's not liquid in its natural state, it's using energies. That's why I call it an active liquid. And as soon as it goes into this dormant state, it goes into a glassy state. It becomes a solid. And when it exits, it comes back into this active liquid form. I want to contend with you that this is what we associate with the normal processes of living, this liquid state. Okay, that's what allows us to function. That's what allows us to move. If I am like a block of ice or wood, I will not be able to move. I will not be able to carry out any of my functions, right? So there's a delicate balance between being a liquid and a solid that really allows us to perform all these functions of life. Okay, so what might a cue be? So let's watch this jamming process in action, right? So here's a paper from 2016 where a change in the pH gives the cell a trigger saying that, now something, trouble is coming. So I'm going to jam up. Okay. Please remember here, we have not removed water, right? So I'm trying to tell you situations of this broad phrase that I've used, without water, right, to mean a transition to go from this state where things are happening active and living to a state where it enters dormancy. Again, this is a movie. Yeah, this will play maybe. Yeah, okay, great. So here are these little cells. And inside the cell, there are these molecules which are glowing green, which are the ones that are tagged. Below is a movie of that process. So you can see that when the medium is normal, these things are just moving around. Right? These are the little jiggly motions of a fluid. At some point, they change the pH. And immediately you saw that everything jammed up. Watch the movie carefully. When they change the pH, it'll come up here in red. And watch what happens here. So now it's in the normal state. There, they change the pH and everything has just frozen in place. They change the pH back and it starts to move. So what I want to tell you guys, so of course they've made a statement here that this transitions of the cell from a fluid to a solid-like state and promotes an entry into dormancy. What I want you guys to take away from what I've said so far is that there's some transition from a fluid-like state to a solid-like state that is associated with the transition from what we might call normal life to this dormant form. Okay, that's one message that I want to give you guys. And this transition, like you saw, might be mediated, of course. If I remove water from this, it dries up. That's an obvious way for you guys to think about entry into dormancy. What I'm telling you here is that there can be another kind of entry where even though there is water, the water is useless for it to do anything because there's no source of energy. There's another way for us to do it. Freeze it. Right? So there's this incredible creature called the wood frog or the Arctic frog. Every winter, its entire body freezes and it becomes wooden. Right? And when the temperature increases, when spring comes, it thaws and it hops away happily. Right? For this entire winter, this frog turns solid. Okay? What exactly is going on there? Of course, water is there, but it's in the form of ice. And again, it's an unusable form of water for this organism right now. There are other creatures like worms, which are a favorite in the lab. Again, these so-called C. elegans. It's a nematode worm, which is found in the soil. This also has this ability to enter dormancy. Okay? And I've used another word over here called cryptobiosis. Secret life. Right? Because we don't know what is going on in this dormant form. It's just code for that. Of course, we are all familiar with the example of seeds. Right? These are dried form with the potential to give rise to another. So the question is, how long can these things remain dormant? That's one thing that we posed earlier. How long? Can it be a year? Can it be 2 years? Can it be 3 years? Here's an example of a paper that came out recently where some of these seeds are buried in ice for 30,000 years. And 30,000 years later, scientists extracted the seed from this permafrost, this ice, and out of it came a healthy plant. Okay? This is insane, 30,000 years. There's evidence for us now to believe that there are some spores, bacterial spores, which can last millions of years. Millions of years, right? That's a staggeringly long amount of time. How or why can these things last that long? That's a very, very interesting question for us to ask. Of course, inspired by things like this, the fact that you can have the potential for life for many years, there are people now that want to put themselves into this state, right? So there are companies in the world that want to freeze people, make them go dormant, and who knows, maybe they want to come back to life. I don't know when. Is this even possible? Is this science fiction? We know that I can't put my finger into the freezer, and when I pull it out, I'm pretty sure it will not be functioning quite the same way, right? With what audacity, with what hope are we going into this kind of an experiment? We don't know yet that this will work, right? But it's a dream. Can it be possible at all? I don't know. So what's going on during this process of freezing? Okay, the water this is another kind of without water, of course. So the cell, so what is drawn over here are molecules which show the boundary of a cell. And when the cell detects that it's going to be frozen, what it does is immediately use pores, ion channels in the cell membrane to get as much water out as possible. So the cell actively says, water, I'm going to try and push you out. Why is the cell trying to push water out? Because if the water remains inside the cell, it forms a crystal. An ice crystal, you have seen ice crystals, they are sharp, right? And these sharp structures can tear holes into the cell. And if they tear holes into the cell is no longer a cell. All its contents are spilled out, right? So that's one reason why the cell is trying to get as much water out, even when it's frozen, remember, right? It starts to produce antifreeze proteins. Why does it care to do this? Because it wants to suppress the formation of ice. Okay? It wants to suppress the formation of crystalline ice. Because crystalline ice is what is sharp. Okay? But I just told you earlier that water need not become crystal. It can also become a solid by becoming a glass. Right? So some of these proteins help this kind of solid formation. Okay? That's what an antifreeze protein does. And it also, the cell also, now, those of you who have studied about proteins, etc. in school, know that a protein is a long polymer and then it folds because of various electrostatic interactions, with hydrophobic interaction. It needs water for it to form a structure, right? But now, this is a state where the cell is getting rid of water. So how does this molecule maintain its structure? Because it has to maintain its structure for it to function at all, right? So, again, the cell starts to make other molecules which mimic water. We don't know yet exactly what these are, how they function, so on and so forth. But what I want to tell you is that the cell starts to go into a completely different chemical composition altogether when it goes into this dormant state. So the question, how long can this thing last? We've just seen 30,000 years, 1,000,000 years. But we know that even some of our most sturdy structures that we build go into ruin if we don't maintain them. Right? So why should something tiny like a cell be able to withstand all these things when something so sturdy as our own constructions will just naturally decay, right? So what it suggests to us is that there must be some maintenance going on, or there is some construction which the cell is able to do that we cannot do, which can last millions of years. In understanding this is interesting from both perspectives, right? So let's take the first perspective. I mean, if we don't go into our room and keep cleaning it, it naturally becomes disordered, dirty, goes into decay, becomes that eventually. So how do we maintain it from not doing that? By spending energy. Right? We go in there, we vacuum it, we clean it, we do all that stuff. We spend energy. So one possibility is that the cell spends energy to maintain itself. Right? So what we are contending with is a situation where on the time scale of 30,000 years, there's enough energy for the cell to maintain itself. So how much energy has it packed for itself? How much food has it carried with it? Right? That's a crazy, crazy thing for us to ponder. But there is evidence to suggest that there is metabolism and maintenance at low temperatures or dormancy. Okay, so what you're seeing here is the metabolic rate measured as a function of temperature, and you see that there is even some growth of organisms that happen at minus 20 C. This is not surprising for some of us. There's maintenance, of course, going on at these very low temperatures. This is energy that is associated with the spontaneous decay of molecules. So the cell at least has to spend that much energy in repairing processes, etc. Right? But we have no clue, we have no hint to suggest that some of this is going on in this dormant state. Okay? Which very quickly brings me to the kinds of questions that we are trying to answer in our lab. We are trying to ask what is happening during dormancy. Okay? What are the ways in which cells are getting into dormancy? What are the ways they're coming out of dormancy? What are the mechanisms which help prevent repair on the way out, or which help prevent damage on the way in, which repair on the way out, etc. But there are very other important questions. How much energy is it packing for itself, like I've been saying? What key processes are running for this maintenance? What are these? Can we figure these out? How long can they keep running? Is there a limit? Can this go on forever? When does the cell give up? When does the cell decide, no? And when it's coming back out, what does it first do? Right? And I'm very happy that the person running all these experiments, my PhD student Charu, is sitting right here. So if there are questions about the next bit of the talk, you know where to go. So she is the person that is running all these experiments, and so she can tell you a lot more than I can even begin to do. Okay, so what is Charu doing? So Charu works with this organism called Saccharomyces cerevisiae. It's the yeast that bread makers use to make bread. These are the cells under the microscope. They are, a few microns in size, a fraction of the width of our hair. Why is this important? It's because we can view them under a microscope. We can visualize these things, the molecules, etc., moving inside, so that Charu can keep track of the motions and see what processes might potentially be going on. And importantly for us, they're genetically tractable. What does that mean? We can go in and make modifications to the gene and play with the processes of life and ask how do those modifications are associated with some of these transitions to dormancy, etc. So this is the very ingenious experiment that Charu came up with. So what does she do? So the question she's trying to ask over here, if I take these cells and I freeze them, they will probably go into dormancy, but not everything will go into dormancy. A lot won't. And they will die, right? A lot will die. But some will survive. So she reasoned, saying that maybe there's something special about the ones that survived. So what if I take those ones which survived and grow them again and keep repeating this process? Can I get cells which become better and better and better and survive this process? So what she does, she grows cells for 24 hours, takes a small fraction of those cells, puts that in liquid nitrogen. For those of you who don't know, that's minus 200 degrees Celsius. We are not putting it into, we're literally dunking it as low as we can go, almost as low as we can go, minus 200 C. Then she brings it back to room temperature. Some of them have survived, like I said. So she measures how many have survived. And then she asks, as I repeat this process, what will happen? And here's what she finds. So here's the percentage of cells that survive as a function of the number of times that she's doing it. It starts off with some 5, 10%, 5% survival. That's a small number, right? She's taken that and gone on and on and on. And after about a month, about 80% of these guys survive. Right? So what you're watching here, is evolution in action. Right? So what we are evolving for, what Charu is evolving for, is the ability to make this transition into dormancy and out of dormancy. The ability to survive without water at this low temperature and back. Potentially, this has the same thing when you dry it. You don't need to freeze it. Maybe you take the same cells and you dry them, make a powder like what we have done outside. Outside, some of you guys who are running the experiment know that not everything hatches. Maybe if we do this, everything will hatch, right? So I'm going to just flash these questions again that Charu is trying to answer. So when these things have this capability, how are they affecting these various things that I raised earlier? Right, so that's as much as I want to say about transitions to dormancy, surviving without water in various interesting scenarios, etc. But I just want to leave you with the message, please stay tuned, because there's a lot going on. We're figuring out how many of these processes work, and if some of you are interested, please join the fun. You can find out more information about us there. So in the lab, what I talked to you about is this work that's going on this transition to dormancy, but there's also another arm where we are trying to use some of the ideas that we learned to construct synthetic systems, which may be of use engineering-wise, or may have the applications outside, etc., but may also lead us to insights about how the non-living becomes the living, right? The origin of life scenarios, and that's the group, and thank you very much.